All right, good morning. I'm Jenny DeLuca, and it is my pleasure to introduce today's speaker, Dr. Tiffany Weir. Dr. Weir has been an assistant professor in the Department of Food Science and Nutrition at Colorado State University since 2011. Dr. Weir received her Bachelor of Science in Biology with an emphasis in genetics and development and a minor in microbiology from Pennsylvania State University. She then continued at Penn State to receive a Master of Science in Plant Pathology before going on to receive a Ph.D. in Cell and Molecular Biology from Colorado State. Dr. Weir has co-authored over 25 articles and book chapters. She has received several honors and awards, including an International Life Sciences Institute Future Leader Award. Dr. Weir also maintains numerous professional memberships and activities, including serving as a horticulture advisor to the Peace Corps Masters International and a consultant for several groups, including the USDA and the Denver Diabetes Foundation. Dr. Weir's research interests focus on the interactions between the gut microbiome and phytochemicals and their effects on human health. Today's seminar is titled Effects of Estrogen Loss by Gut Microbiota and Potential Mitigation by Administration of Oral Phytochemical. Please join me in welcoming Dr. Tiffany.
Thank you, and I'd like to thank Dr. Turner for the invitation to get to come in and speak with you guys today. I did pare down my title a little bit. That was quite a mouthful. I'm not sure what I was thinking when I sent it. But what I'm going to talk about today is a little bit broader than just the phytoestrogen mitigation. We are examining just the effects in general of estrogen loss and its effects on the intestinal microbiota and intestinal dysbiosis. And in terms of mitigation, we're not only looking at phytoestrogens, but actual estrogen replacement therapies, exercise, and different factors that might help mitigate some of the effects of the intestinal dysbiosis. So the problem that we're interested in investigating is that menopause, which is the end of a woman's reproductive life, is characterized by altered hormone levels, which includes a decrease in estrogen levels. Now, it's not a complete loss of estrogen. There's still estrogen that's produced from adipose tissue from conversion of androgens. So there is some estrogen still, but it does decrease significantly at menopause. And postmenopausal women are at a much higher risk of developing type 2 diabetes, cardiovascular disease, and certain types of cancers. Generally, in terms of physiological changes to the body, the adipose tissue deposition shifts and it goes from the subcutaneous or the pear-shaped adipose tissue deposition more to the apple-shaped or visceral adipose tissue. So some of the effects of postmenopause are thought to be due to greater inflammation in visceral organs due to this shift in adipose tissue deposition and increase in central adiposity. We also think that this increase in central adiposity is associated, or either perhaps the shift in fat deposition is caused by or exacerbated changes to the gut microbiota. So a lot of these conditions, cardiovascular disease, type 2 diabetes, and some cancers are also associated with shifts in the gut microbiome. So the problem that we want to explore is, does estrogen loss affect the composition and function of the gut bacteria populations, potentially contributing to intestinal dysbiosis that's associated with some of these diseases post-menopause? So a little bit of background in the gut microbiome, and we won't go into too much detail here because I know that you guys have heard talks on the gut microbiome before, but there are approximately 10 bacterial cells in our body to every single eukaryotic cell that we have, so we are more bacteria than human. Obviously, they can't just be hanging out there without some sort of contribution to our host physiology. There are roughly 100 times more bacterial genes that are encoded for than our human genes. And because of that, our gut bacteria help us carry out a number of functions that our body wouldn't be able to carry out without these bacteria. They include things like assistance with digestion. So carbohydrates and proteins that don't get digested in the stomach and make it into the colon are then fodder for fermentation by our gut bacteria. One of the primary products of this fermentation are the short-chain fatty acids, which include butyrate, propionate, and acetate. In general, these short-chain fatty acids are thought to be beneficial. They can help with appetite regulation, glucose regulation, as well as butyrate is the primary energy source for the colonic epithelial cells, but they've also been associated with obesity when individuals are in states of overnutrition. They also are important for pathogen exclusion. So our bodies, we consume lots of different foods. We encounter, different bacteria when we're consuming these foods and viruses and things like that, but we rarely get sick from them. The reason is that either our stomach acids take care of them, but if our stomach acids don't take care of them, and they do make it into the small intestine, then we have our commensal organisms that are there that are going to protect against these pathogens from being able to colonize. They do this through a number of different mechanisms. Sometimes these are direct mechanisms, like the production of antimicrobial compounds that's directly going to wipe out these pathogens. It can be through creating microenvironments, areas of high acidity through production of lactic acid and things like that are unfavorable for the pathogens to be able to survive. They can take up all the good nutrients. They can take up all the best real estate, so there's really nowhere for these guys to colonize. And then they also can stimulate host immune defenses. They'll stimulate the secretion of mucins in the intestines, as well as regulate immune and inflammatory responses that can help protect against pathogen infections. So our gut microbiota changes pretty significantly throughout the lifespan. We've really studied a good deal what happens at birth and early infancy, because we think that these are important in the development of tolerogenic responses, and that improper colonization in early life could lead to problems later on in life in terms of developing autoimmune disorders and food allergies and other types of issues. So we know that in infants, the gut microbiota is highly dynamic, it changes constantly, and it really doesn't start to stabilize until about 1.5 to 2 years of age with the introduction of the solid foods and the typical diets. Now, from that point on, it starts to stabilize and it starts to look like an adult gut microbiota. And throughout most of the adult lifespan, it remains relatively stable. You can have changes due to antibiotics, stress, diet obviously is one of the biggest factors that's going to induce changes in the gut microbiome. But a lot of times these shifts are going to be small shifts, not in the whole composition of the global profiles of microbes, but in individual organisms. And then when you have disease states, you'll also have shifts in the gut microbiome. We'll talk a little bit more about that later. It's a term called dysbiosis. What we don't know a lot about is what happens here in later life. There have been some studies done that suggest that the gut microbiota starts to shift again, perhaps after the age of 65, although again, there have been relatively few studies and they're on relatively small cohorts. So it's difficult to pinpoint the exact age when that shift occurs again. There have been studies suggesting that the health status of an elderly individual is going to determine whether or not those shifts are dramatic. Healthier individuals, again, are going to tend to have gut microbiota that look more like the microbiota that they had when they were middle-aged or younger. And then individuals who are more feeble or ill are going to have larger shifts in their gut microbiota. But we do think that there are some changes that occur with aging, and we don't really know what causes those, if these are environmental factors or if these are genetic factors. But we do think that hormone changes that occur in later life might be one of the causes for these shifts in the gut microbiome. So intestinal dysbiosis is what happens when you do have a pretty dramatic shift in the gut microbiota. It's usually characterized when you have a nice healthy microbiome. You have a good diversity of different types of organisms that are present. The colonic epithelial cells are nice and healthy, and the junctions between them are tight, so it's only allowing nutrients and regulated things to pass through and not just open for bacterial effector molecules and bacterial translocation. And then there's a nice healthy mucus layer that's present that protects the epithelial cells from the luminal contents. And then an event can occur in this particular diagram. It's an inflammatory event, but in the case of what we're talking about today, this could be an event such as a shift in hormone levels, like loss of estrogen. And in our animal models, it does tend to be an acute event because we do ovarectomy, so we remove the ovaries, and all of a sudden the estrogen levels have changed overnight. Now, obviously, in human populations, it doesn't occur like that. for the most part, it's a more gradual change and a more gradual shift in hormones. So it might not be as dramatic as this, but over time, it can lead to this state of dysbiosis. So the dysbiosis here is characterized by a lower diversity of organisms present, inflammation in the colonocytes, a breakdown of these tight junctions between the cells, and a degradation of this mucus barrier. This is just showing a close-up here of the tight junctions between them. So there are proteins that are produced in the nucleus and then translocated, primarily zonulins and occludins, that form these bridges between the epithelial cells, and then they can regulate what passes through. So when that barrier system breaks down, what can happen is that you can get translocation of bacteria and bacterial effector molecules that can lead to more systemic inflammation. So dysbiosis of the microbiota has been associated with a number of different chronic diseases, including cardiovascular disease, dysfunctions in metabolism, including development of type 2 diabetes, non-alcoholic fatty liver disease, certain types of cancers. Within the intestines themselves, you can have inflammatory bowel diseases that are associated with changes in the gut microbiota. And then some more autoimmune type things like psoriasis in the skin and skin allergies, as well as rheumatoid arthritis and joint issues. And this is when you have that breakdown in the gut barrier function, you can get bacterial effector molecules. The one that's been the most studied is lipopolysaccharide. So the lipopolysaccharide can interact with receptors on different immune cells and other tissues and cause a low-grade inflammatory response. And so we think that it's this low-grade inflammatory response that at least partially contributes to the development of these different conditions. And this is just some data over here that's showing in colorectal cancer differences that are present in the gut microbiota between individuals that are healthy and individuals who have colorectal cancer. Now, there have been a number of studies that have been done in healthy individuals that have tried to identify what they call a core microbiome. And it's been really difficult because of the variability from person to person to come up with this core microbiome. So for the most part, dysbiosis is defined in a case and control type situation where you have individuals that have a particular pathology or disease and then individuals who do not have that. And so that's how we've been able to define dysbiosis, because we haven't been able to define a healthy gut microbiota. And so in this case, there were actual taxonomic changes in the gut microbiota that were associated with colorectal cancer. This is a little bit of a gratuitous slide, but I wanted to throw in some of my own work up here. We didn't see changes in the overall global profiles of the gut microbiota between our colorectal cancer patients and healthy individuals, but what we did see was a significant shift in the metabolites. So occasionally, instead of seeing changes in the profiles of the bacteria that are present, you're going to see shifts in the functions that those bacteria are carrying out. And so, studies that are combining microbiome and metabolome are really starting to get at some of these mechanisms a little bit better, rather than just leaving them at associations between the diseases and microbiome changes. So shifting gears a little bit to talk more about estrogen, why do we think that estrogen and loss of estrogen might be having effects on the gut microbiota? Well, estrogen has receptors in the body. There are two: estrogen receptor alpha and estrogen receptor beta. They're found in different tissues, and they have very different tissue-specific effects. But we know that in the colon, you find an abundance estrogen receptor beta. And there haven't been that many studies done to determine what estrogen receptor beta does in the colon. It was only identified as a separate type of estrogen receptor, I think, in the late 90s. But the few studies that they've done, they've looked at expression of colonic ER beta, and they found that it's inversely associated with colorectal cancers. Also, if you give estrogen treatment or estradiol, it acts through ER beta to induce apoptotic activity and prevent initiation and promotion of colorectal cancer. So if you're giving estradiol to cancer cells and cell lines that have already developed, there don't tend to be any effects. But if you give them early on, they might actually be more preventative because they're stimulating apoptotic and regulating the growth of the healthy cells. And then ER beta stimulation has also been shown to improve barrier function in the intestinal epithelial cells. So they've also done some monolayer studies with different cell types, and they found that when you stimulate ER beta with either estradiol or other estrogen receptor agonists, that you have more resistance in those barrier cells, so better barrier function. And also they've looked at biopsies of patients who have IBD and found that there's a lower expression of this estrogen receptor beta in people who have chronic inflammatory intestinal diseases. So what about the evidence to suggest that ER beta has some influence on the composition of the gut bacteria? And this was a study that was done out of Texas A&M here, a group that Dr. Storino, but he looked at mice that were knockouts for the estrogen receptor beta. And what they found is that in the absence of ER beta, there were significant differences in the species richness of the gut bacteria. So just the lack of that receptor alone without any changes to diet or any other parameters caused a significant change in the composition of the gut microbiota. Now, to complicate matters a little bit more, this change was altered by giving the animals different diets. So in this experiment, they also gave them, this was a phytochemical rich, complex diet. And then this was a highly purified and processed diet over here. So what you can see is that you have a difference between the ER beta knockout animals and the animals that have ER beta in both cases, but the actual species richness shifts depending on which diet they're on. And this is really interesting because it reflects some of the results that we've been getting. And so there is an effect of ER beta on the microbial population, but it seems that diet might actually be more important. So diet could trump that effect potentially. So this is showing here, I think this is the phyla level breakdown of the gut bacteria in the two situations. It's just demonstrating the differences in the composition. You've got the ER beta animals over here, ER beta minus and ER beta positive animals up here. And you can see that there are subtle shifts in the overall distribution at the phyla level of the bacteria. But then there are larger shifts here as you shift the diet the animals are on. So this was a study that was done in humans, Dr. Furman's group, and they were interested in looking at how estrogen metabolites in the urine correlated with the complexity of the gut bacteria. And so they had three different populations. They looked at males, they looked at premenopausal females, and they looked at postmenopausal females. And they found in the case of males and postmenopausal females that there was a correlation between the amounts of urinary estrogen and the species richness of the gut bacteria. So the more estrogen there was, the more diversity there was in the gut bacteria. And this was associated with increasing estrogen levels, were correlated with increases in bacteria in the phylum Firmicutes, particularly those that are related to the Clostridiales. So based on the accumulation of all this different evidence that the gut bacteria and loss of estrogen lead to similar risks in developing certain types of diseases, that we have estrogen receptors in the gut, and that those estrogen receptors are important in intestinal barrier function, and that they seem to also have an effect on the composition of the gut bacteria, we've developed a set of research questions that we're interested in exploring. So the most basic question is just, is the composition and function of gut bacteria significantly altered when estrogen is removed? If so, do these alterations lead to a pro-inflammatory intestinal state? So just because the gut bacteria are changing, that doesn't necessarily mean that they're changing in a negative way. So we're interested in looking at whether these shifts that may occur with estrogen loss are positive or negative. So we're going to measure things like characterization of the epithelial barrier integrity, whether or not we can find levels of metabolic endotoxemia, so the LPS that I mentioned that can translocate and interact with receptors like the toll-like receptors and the NOD-like receptors. We want to see if we have activation of some of the, or increases in some of the cytokines, as well as whether or not we have increased levels of endotoxin or LPS in the blood. And then also whether it alters profiles of microbial metabolites. We're particularly interested in looking at some of the metabolites of estrogen itself, because there have been some studies done, the same group that looked at the characterization of urinary estrogens in the gut bacteria has also shown that there are different ratios of the parent estrogen metabolite to some of its conjugates, and that depending on those ratios, that alters the risk for developing breast cancer. So we want to look at some of the estrogen metabolites. And then how do exercise, diet, and estrogen replacement, either as synthetic estrogen or as phytoestrogens that can be found in foods, affect intestinal changes that are associated with, and that should say estrogen loss, not intestinal loss. And then again, finally, the last question that we're really interested in is the timing aspects of this. And so menopause in women is a process, and it occurs over several years. And we want to see whether or not there's an optimal time for intervention. If you have to start intervening with estrogen replacement in perimenopause, if early menopause is a sufficient time for intervention, or can you reverse some of the effects if you do these treatments in late menopause? And so that's an area that we haven't really gotten into in terms of the work that we're doing yet, but that's a direction that we're moving in. So these are some of the current studies that we have ongoing or completed. The first study is the study that we've completed, and I think the paper was sent out to everybody to read, but I'm going to go ahead and just kind of summarize some of our main findings from that. That was in a study with rats that have inherent high or low aerobic capacity. These are animals that were identified and are bred in Michigan. And basically they found that in the absence, they're not transgenic animals, it's a polygenic trait that's conferred that alters the ability of these animals to run different distances. So the low aerobic capacity and high aerobic capacity are classified according to their running distances. And what's really interesting is that they can pass that trait along. So when they breed these animals, they found that their offspring fall into either the low or high aerobic capacity groups as well. And this was really kind of a serendipitous study for me, because we had somebody in our department who was looking at cardiovascular disease and endothelial function. He got these animals in, he did some pulse wave analysis and didn't see any differences in them and said, I have these, does anybody want them? Do you want to do something with them? And another one of my colleagues is interested in whether or not exercise alters the effects of estrogen loss. And so she said, well, they're all females, why don't we overrectomize them? So I ended up just collecting stool samples and not knowing what or if I was going to come up with anything interesting. And it turned out that we did see some interesting effects in terms of changes to the gut microbiota. The other two studies that I'm working on currently are still pretty early stage. We've actually conducted the studies in the animals, but we are still in the process of looking at the data. So I have a little bit of data to show from those. It's still very kind of rough data, but there are some neat things that we see coming out with that as well. So I'm going to go ahead and show some of that too. But one of them is in mice that have been overrectomized and then fed a high-fat diet. The rats that we used were on a highly purified phytoestrogen-free diet that was a maintenance diet, so for weight maintenance. So we decided that we wanted to see what would happen if we had animals that were on a high-fat diet group. And then the last study, and this is the only study that we've actually kind of intentionally performed, was taking animals and overrectomizing them and then seeing what happens to the gut bacteria when we supplement them with either replacement estrogen, so we give them estradiol, or we give them an extract from hops, which is also rich in phytoestrogens, to see if we can return gut homeostasis in the treatment groups. So the first study was looking at the relationship between estrogen loss, aerobic capacity, and the gut bacteria. As I mentioned, this one's already completed, but we had the two groups of animals, the high aerobic capacity and the low aerobic capacity animals. And so we had one of the groups of each that we conducted sham surgeries on, as well as another group that we conducted ovarectomies on. Now, these were already older animals. Like I said, they'd been used for other experiments and had been kind of hanging out in our animal facility for a while, which was kind of nice because this allowed us to look at animals that were actually sort of more relevant to a human population. We did the surgeries and we maintained them on their phytoestrogen-free diets for 9 weeks after the surgery. And then we measured a lot of metabolic parameters because that was what my colleague was primarily interested in. And what we found was that the ovarectomy resulted in increased body weight in the animals, which was expected, and decreased uterine weight, which was actually our indicator of whether or not our surgeries were successful. And so this was all expected, but the HCR were actually moderately protected against the OVX induced weight gain. So they did gain weight after surgery, but they gained less weight than the group that were in the low aerobic capacity. What is also interesting, though, is that we didn't find any metabolic dysfunction in any of the groups. So they hadn't gotten to the point yet where they had any insulin resistance or dysfunction in their glucose metabolism. We also measured liver triglycerides. So that's sort of an early stage indicator of metabolic dysfunction, because before you're going to have insulin resistance, a lot of times you're going to start accumulating lipids in the liver. And we did see that there was a significant increase in the liver triglycerides with the low aerobic capacity overectomized group, and that the high aerobic capacity group was protected against this accumulation of liver triglycerides. And then we decided to look at the gut bacteria. So this was really kind of my area where I came into these experiments. And what we found was that the sham animals, whether they were high or low aerobic capacity, didn't have significantly different gut microbiota. But after they were overectomized, there was a difference between the sham and the overectomized low aerobic capacity group. And that change was characterized by a decrease in the Firmicutes and an increase in the Bacteroidetes. Now, there are a number of different associations with those two major phyla. What, in general, the Firmicutes, the higher Firmicutes to Bacteroidetes ratios are associated with obesity. But in this case, we actually saw the opposite shift. Now, the Bacteroidetes are primarily gram-negative organisms, and so those are the ones that produce the lipopolysaccharide. So what we think here is happening is that there's the potential for greater inflammation because there's higher levels of the lipopolysaccharide, at least in the luminal contents. So if there is dysfunction of the gut barrier, then there's the potential for more translocation and generation of inflammation. There was no significant difference between the HCR overectomized and the HCR sham groups. But what's interesting is that taxonomically, again, we didn't see a shift in the gut microbiota. At the species level, there were some changes in all the groups, and those are characterized in the paper. But when we looked at the short-chain fatty acid production, there was a significant decrease in short-chain fatty acid production in the HCR group. So even though we don't see the taxonomic shifts, again, we're seeing a somewhat potentially unfavorable metabolic shift in terms of the production of those short-chain fatty acids. So conclusions, the HCR rats were modestly protected against increased adipose tissue and liver triglyceride accumulation in response to overectomy compared to our low aerobic capacity animals. The global microbial profiles were not significantly different between the HCR and LCR shams, but the OVX was associated with global changes in the microbiota of the LCR animals, including increased bacterial diversity and a shift from the Firmicutes and Bacteroidetes ratios. And although the microbial composition didn't change with the OVX HCR, we do start to see some change in the function of those microbes. So again, the fermentation capacity of those organisms changed with the evident with the decrease in the short chain fatty acids. So the next study, again, this was we decided to change our technique for the ovarectomies. We were going in the front and the animals would tear at their stitches and cause themselves additional stress. So we shifted to a dorsal entry ovarectomy. And we wanted to practice, so these were some leftover animals that we decided to practice our surgical technique on. And because we didn't kill them when we did the surgeries, we decided to keep them and just put them on different diets to see what happens. So this is one that would need to be repeated because our numbers are pretty low, but we just wanted to see what kind of data we could get out of these animals. And so we had again, the OVX and the sham groups. One group maintained on the chow diet, and the other group was shifted at surgery to a high-fat diet. And we saw that the body weight gain was higher, again, in the OVX group. Again, this is expected after OVX. Even in the chow, when they're maintained on the same diet, they do tend to gain weight after ovarectomy. We did, again, see greater weight gain overall in the high-fat diet group. And we saw significantly lower food intake in this group, though. So when we looked at the gut bacteria differences, and again, this data's, I just got this sequence data back about a week ago, so I haven't had a chance to look at some of the parameters like the microbial diversity and the richness, and some of this I've only really looked at what's going on at the phyla level at the moment. But at the phyla level, we see that the ratio of the Firmicutes to the Bacteroidetes is changing again. And in the sham and OVX groups that were on the chow, those ratios shifted in a similar way to what we saw with the rats that were on the phytoestrogen-free diets, in terms of we have an increase in the Bacteroidetes and a decrease in the Firmicutes. When we have them on the high-fat diet, however, we see that there's also a shift in those profiles, but it occurs in the opposite direction. And so that sort of reflects the paper that I discussed earlier where they had the ER beta knockout animals, but they put them on the two different diets. So we're seeing effects from the loss of estrogen, but those effects tend to be trumped by the change in the diet. So the high-fat diet's effects are stronger on the gut microbiota than the effects of the hormones, it appears to be. And then the last study, this is where we wanted to look at what some of the interventions that we can do might, including hop extract. So the hops I'll talk a little bit more about, but hop extracts rich in phytoestrogens. And then we did estradiol treatment as well. So again, we have the two surgery groups, the OVX and the sham group. And then in the OVX group, we did a placebo, the hop extract, and the estradiol. In the sham group, we did just the placebo and the hop extract. We dosed them using cookies because we didn't want to be gavaging these guys every day. And I don't know, any of you drink IPAs? You know what the hops taste like? It's pretty bitter, and when you purify out the hops and extract that, it's almost unpalatable. So we were going to try to put it on the animals and have the animals, they'll groom when you put something on them. So we wanted them to try to lick it all off, but they didn't like it, and they'd get a couple licks in and they'd kind of be like, oh, this is nasty. So we found a paper where they used Nutella to hide the taste of the hops extracts, and we just working with the Nutella and dosing it had some compounds in it that we didn't want in the Nutella. So we found these quadratini cookies, which were actually like expensive imported European cookies. So these were very well treated animals. They got fancy cookies every day. And that's how we got them to consume their hops extract. And then we put the estradiol on them as well. Everybody got cookies. So everybody was treated fairly. These particular animals were already 7 months old when we ordered them. They were retired breeders, and it was probably another maybe month after we got them before we actually conducted the surgeries on them. And then we followed them out for 12 weeks. So these were the oldest animals, the oldest mice that we used. The rats were also pretty old. But they were already sort of nearing, natural end of their life by the time we did the termination of the experiment. And I bring this up now because I think that it may have some bearing on the results that I'm going to show you in terms of the microbiota. First, we'll go into a little bit of background about hops and why we chose hops. Colorado State University's nutrition department is kind of like you guys. We have nutrition and food science, but our food science is focused on fermentation science. And so we're very interested in working with the fermentation industry. And one of the issues that our brewers have is that they have all the spent hops. And so we would like to find ways that we can get that spent hops and use it as a value-added product. So the spent hops, the alpha acids that give the flavor to the beer, are extracted during the beer brewing process, but the phytoestrogenic compounds are still there in the spent hops afterwards. Now, we didn't extract our own compounds. We actually went to Metagenics and we got a product that they market in Europe that's called Menohop. The actual Menohop product has soy in it. So we had them give us the version that didn't have soy. It just has these hops phytoestrogens, but that's why we decided to use hops. And the phytoestrogenic properties of hops were first noted when they realized that women hop pickers, their cycles would start to shift once they were picking the hops. It was actually a very potent phytoestrogen, so it was absorbed through their skin. And so they started looking at some of the compounds that were present in the hops. The primary compound is called xanthohumol, which spontaneously will isomerize to isoxanthohumol. So those are the primary things that you're going to find in the hops extract. And neither one of those things had phytoestrogenic activity. But what they found was there were also very low levels of this other compound called 8-prenylnaringin. And the 8-prenylnaringin does have very strong estrogenic activity, but it's fairly low in the crude hops extracts. However, there are gut bacteria that when you consume the xanthohumol or the isoxanthohumol, they will convert those compounds into the 8-prenylnaringin. So depending on the composition of an individual's gut bacteria, they're going to be more or less efficient at producing the 8-prenylnaringin compound. And there have been some studies in humans that are characterizing the differences between people, and most people fall into one of two categories. They're either high or low 8-prenylnaringin producers. And just for you guys who might be wondering, there are no appreciable amounts of this in beer, so go ahead and drink the IPAs. It's not going to hurt you. So because we were orally dosing, we wanted to make sure that these compounds were actually getting into the animals. So we did some pharmacokinetics to determine whether or not we could detect the 8-prenylnaringin and the xanthohumol in the blood. And because of the cost of the metabolomics, we collected time points 1, 2, and 3, but we only started doing the metabolomics at 4 hours because previous studies had shown that that's where there's the peak amount of phytoestrogens that can be detected after oral ingestion. And that agrees with our results. We are going to go back and look at the earlier time points to make sure that it's not accumulating higher in an earlier time point. But we see the 8-prenylnaringin and the xanthohumol here. These are on different graphs because they're different scales. We actually had standards so we could quantify these, and these are based on mass and retention time, but we didn't actually have standards, so they're only relative abundance. We also saw that there are a number of 8-PN glucuronides, so conjugated 8-PNs. We don't know how that affects their bioactivity. So when we looked at the body weights of the animals, we saw that the hops had absolutely no protective effect on the weight gain after ovarectomy, which was initially a little bit disappointing, but now that we think about it, a lot of the results that we look at downstream could be explained by weight loss or lack of weight gain. So it actually works out better for us that our hops animals weren't protected against weight gain, because that means that any effects that we saw are independent of changes to the body weight of the animals. In terms of the subcutaneous and visceral fat accumulation, you can see that there's a little bit more visceral fat accumulation, what I talked about in the beginning, that central adiposity, in the animals that were just overectomized and not treated, or the animals that had overectomy and HOPS. So we still see more central adiposity. But in the HOPS group, we did see less overall total adipose tissue relative to their total body weights. So it was somewhat protected against overall fat gain. And just to point out the estrogen treatment here, no weight gain relative to the sham groups in the estrogen group, and same with the fat accumulation. They actually had very similar profiles to our sham group. So what we did see, though, when we looked at the liver triglycerides is even though there wasn't really protection against visceral adiposity and there wasn't protection against weight gain with the HOPS extract, we do see that there was protection against accumulation of the liver triglycerides in this group. And so here's our OVX placebo. The HOPS extract significantly reduced that over the placebo group, but it was also similar in terms of the amounts of triglycerides that were present in our sham animals. And then again, the overectomized group with the estrogen treatments were very well protected. Now we went in to look and see what might be associated with that protection that's present in our HOPS extract. And the xanthohumol has been reported to lower liver triglycerides via SREBP regulation. Also, metabolic endotoxemia and gut dysbiosis is associated with fatty liver. So we were hoping that we would see some protective profiles in terms of intestinal inflammation, which we haven't looked at yet, but we are planning to. So again, this sequence data just came in with the other sequence data. And when we looked at the differences between the groups, surprisingly, there were absolutely no differences. Even the overectomy didn't result in any shifts in the phyla level. Now, we again haven't looked down at any of the lower taxonomic levels, and we haven't looked at the things like the diversity and the evenness and the richness of the populations. But at the phyla level, there were no significant differences in any of our groups. And that was, as I said, sort of surprising. We at least expected to see differences between the OVX and the sham animals because we've seen those now in the other two groups of animals. But I mentioned that this was one of our oldest groups of animals. So these were mice, and they were already they were already retired breeders, which suggests that they may have had some sort of dysfunction in their estrogen metabolism already that caused them to be retired from breeding. And by the time we terminated these animals, they were already over a year old. The typical lifespan's about a year and a half. So when I looked at these profiles, the profiles overall were completely different than the healthy profiles that we've seen with the other animals. We typically see close to 80% of the firmicutes and between 10 and 15 of the bacteroidetes, and then the rest are made up by other types of organisms. In this case, we're barely seeing, or not even seeing, 50% total firmicutes. The bacteroidetes populations are much higher. Now, this is similar to what we saw with our OVX groups. When we OVX'd them, we saw increases in the bacteroidetes in the absence of a high-fat diet. And these guys were not on high-fat diet. They were all on a purified phytoestrogen-free maintenance diet. We also saw this group of bacteria here, which I haven't really been able to find a lot of information about, but one report that I did find said that this particular group is associated with aging, and then higher levels of these verruco microbia. So what I think is happening here is that we are seeing an aged bacteria profile. So we maybe waited too long with these animals to start doing our different interventions and treatments, and that there's something independent of the hormones that is causing sort of that shift that's associated with aging. But obviously, this is something that we need to explore a lot further. So where are we going to go with all this? I need to go back and continue the examination of our microbiota data and look at things like the bacterial population evenness and richness. I think the one study I showed you with the ER beta richness was changed with the gut bacteria. In the human study, it was the diversity that was changed with the estrogen metabolites. So these parameters could be important, and we need to go back and evaluate that. We're also going to start looking at some measures of microbial function and intestinal inflammation in our different treatment groups. Right now, I think this week my students are supposed to be running the short chain fatty acid analysis to look at that. We are also measuring circulating endotoxin levels. We did do what's called phytidextrin with these animals pre-termination, at least the group that had the intervention treatments. And that's where you feed the animals a sugar molecule that's attached to a fluorescent dye, and then you measure the fluorescence in their blood later to look at intestinal permeability. And there were no significant differences between the groups in intestinal permeability. But we did this about midpoint through the experiment, so we don't have the termination measures. We're going to look at cytokine panels and things like that. And then also, we would love to be able to extrapolate this to human populations because ovarectomy obviously isn't the best mimic for natural menopause. It's, kind of hitting them with a hammer in terms of the estrogen loss, and that's not the way things really go. So we have a proposal out right now to look at the gut bacteria composition and different estrogen metabolites in a group of pre- and postmenopausal women. So I want to thank my collaborators, especially Dr. Kim Cox-York. She was the one that kind of started this whole thing with the LCR and the HCR animals. And she's the endocrinology specialist. So I really just kind of keep with the microbial stuff, and she helps guide me through with the estrogen metabolites and estrogen receptors. These are some of my other colleagues in the department who have been involved in this work. Britton and Cook are the folks at Michigan that provided us with the LCR, HCR animals. And then Jay Kirkwood is our metabolomics specialist that did all the pharmacokinetic measures. My grad student, Allie Ham's the one doing the phytoestrogen work. Amy Shefflin did the bacterial analysis for the LCR, HCR, and these are some other members of my lab that helped with animal care and terminations and things like that. And we were funded from Hatch Fund's Colorado Ag Experiment Station, as well as a mini grant from our college. And we got our phytoestrogens from Metagenics and Hopsteiner. Thanks. Any questions?
Open the floor up now. Any questions? Turn on the lights. Maybe I could start by just asking a technical question, because microbiome work is clearly very challenging. But I was wondering what you do with regard to the maintenance of your colony, or tell us about your concerns about the background of the animal and how that could affect things. How you prepare the diets in any way. Like you could be adding hops, but they've got microbes all over them, and they're going directly into your diet, so you're feeding microbes to animals, and another group you're really not feeding microbes. Well, what do you do to sort of address all these confounding variables and feel like you have interpretable outcomes as a result?
Yeah, so we order our animals all from Charles River, so we get them in and generally conduct the experiments pretty quickly afterwards. But they're all, most of these guys have all been individually housed, but in the same room and everything. So should minimize the differences. We use the same age animals for within an experiment, but as you can see, different aged animals, and we think that the different aged animals that we've used in these experiments could be a confounding effect. And it'll be interesting to dig more into the literature and see, most people will only use, 8-week animals, 12-week animals in their experiments. You don't see very many experiments where you're using 7- and 8-month-old animals, partially because they're so expensive to buy. Although these ones, because they were retired breeders, were actually cheaper. And so, yeah, certainly I think, the age of the animals is going to have an effect as long as you're aware of that and kind of take that into account when you're looking at the gut microbiota. And then just normalizing treatments across. So, not everybody got the hops extract, but the hops extract itself, where they came in capsules, they were prepared in the lab, filter sterilized, and then added onto the cookies, and everybody got the cookies, and everybody's food was kind of sitting out in the same environment in the same cages. So hopefully there's not too much additional contribution, but generally the organisms that are consumed get broken down and killed by the stomach acids anyway. So you can detect some of the organisms that come directly from food, but they're usually a very small proportion of the population. Did I answer the question?
Tiffany, I was interested in your comment that your protein fatty acids increased and what should be your protective food. Have you done anything to monitor, say, either absorption or expression of some of the key transporters?
We're looking into that. We want to see if we can kind of characterize the absorption. I mean, well, in this case, we're looking at cecal, short chain fatty acids rather than fecal, so that minimizes loss to the colon, particularly of the butyrate. So that's with the animals ideal. Now with humans, you can't measure. You have to measure fecal short chain fatty acids, and so I think that is really important to take into consideration absorption. But we're looking, and maybe I should chat with you a little bit about looking at measuring gene expression of some of the butyrate transporters.
I was just actually stunned that somebody else was having the same effect with some of our sorghums. The most protective ones seem to be just cutting short chain fatty acids.
Isn't that? Yeah.
And so,
Maybe they're relevant.
yeah. I mean, it's a very intriguing observation. So the question is, here's reducing them or removing them.
Absorption of them. Yeah, I've seen some people are measuring plasma levels of short chain fatty acids now, but I don't know that there's a standard for suggesting, what that actually means, especially with butyrate,
Right.
because most of your butyrate's going to disappear in the colon and not be detected in the plasma. So. Yeah.
So with the animal, so you have the OX animal, then you have the treatment with the estrogen or the how extra you see the decrease in the fat liver or the fat accumulation.
Triglycerides.
So that could be some effect from intestine or could be some direct effects. Have you ever tried either E2 or the